Good day, everyone. Good afternoon. My name is Carlo Lapid, Senior Bioinformatics Officer at PGC's core facility for bioinformatics, and I will be your moderator for today. Welcome to GXBCon 2021, Genomics and Bioinformatics Conference. Thank you for joining us today. Our theme for this conference is Overcoming Challenges, Building Opportunities. And we're excited to introduce to you all of the distinguished guests that we have invited to speak on this topic during this 3-day event. For your friends or colleagues who were not able to register, we're also live on Facebook at www.facebook.com/phgenome. You may also find the live stream link in the Zoom chat. And now, to formally open this event and give her welcoming remarks, it's my honor to introduce Dr. Cynthia P. Saloma, Executive Director of the Philippine Genome Center.
Good afternoon, everyone, and thank you for joining us today. For those of you who have joined for the first time, we hope that you regularly visit our website or follow us on Twitter, Facebook, and LinkedIn as we normally announce our upcoming activities or new updates in these platforms. For those who always join our capacity building activities like webinars, virtual or face-to-face conferences, thank you for the continued interest in the scientific information we share. The impact of the COVID-19 pandemic was felt by everyone, be it physically, financially, mentally, or emotionally. No matter what sector or demographics we are in. With the enormous challenge of the SARS-CoV-2 virus, it is natural to feel despair and disappointment too. However, we can choose to view these times with proactive and calculated optimism. Speaking to you today as a molecular biologist and as PGC's executive director, I urge everyone to discern and make use of these opportunities that the pandemic has brought to us. Sequencing an organism or conducting whole genome sequencing of samples of nasal swabs for certain locations all over the country provides a whole gamut of information which could usher in a dialogue or interaction among scientists and researchers with experts of other disciplines. It is not every day that the mere mention of whole genome sequencing becomes a topic of our national dialogue. This 3-day conference was conceptualized even before the different variants of the virus came into being, with the intention of sharing relevant information to promote an understanding and appreciation of the sciences or the science behind the discussion among data scientists, researchers, and clinicians, as well as administrators as the government manages the COVID-19 situation in our country. Consider the next 3 days as learning opportunities from genomics and data scientists, researchers, and practitioners. Esteemed scientists and colleagues from Luzon, Visayas, and Mindanao, as well as our collaborators from other parts of the globe, will be sharing their time and expertise with us in this 3-day conference. The first day will focus on health topics, while the second day will focus on agriculture, livestock, and fisheries. On the third day, it will focus on biodiversity, ethnicity, and forensics. I hope you all navigate, learn or relearn, and continue to thrive in the midst of this pandemic. As a fellow researcher, let me share that the chance to provide scientific information to the Department of Health and the Interagency Task Force on COVID-19 variants and be part of an evidence-based decision-making at this time of national health emergency is a duty we at the PGC are grateful for. To the dedicated teams in PGC and the panel of speakers behind this conference, thank you and good luck to all of us. Maraming salamat po.
Thank you very much, Dr. Saloma, for those very kind words. To all of you, as Dr. Saloma mentioned, the GXB conference is divided into three selected fields of application. For today, we have health and medicine. For day 2, tomorrow, it's agriculture, livestock, and fisheries. And for the third day, we have biodiversity, forensics, and ethnicity. So today, we open the conference with topics related to health. It's my pleasure to call upon Dr. Eva Maria Cuchonco de la Paz, Director of the Health Program of the Philippine Genome Center, to present her keynote address. Welcome, Dr. de la Paz.
Good afternoon, and thank you for the opportunity to present the Philippine Genome Center's health program and how we tried to overcome challenges and build opportunities to use genomics for better health. This is my disclosure statement. In this presentation, I will provide information on the strategic engagement to identify future research priorities and opportunities in human genomics to improve human health. This is followed by a discussion on the opportunities in relation to the health genomics landscape in the Philippines, and share with you the R&D of the Philippine Genome Center's health program, our current challenges, and future directions. We have witnessed an increasing amount of studies that demonstrate how genomic information can be effectively used in clinical care. It is anticipated that in the near future, the advances in technology development, biological insights, and clinical applications will lead to more widespread integration of genomics into almost all areas of biomedical research, the adoption of genomics into mainstream medical and public health practices, and an increasing relevance of genomics for everyday life. The US NIH National Human Genome Research Institute recently completed a strategic engagement to identify future research priorities and opportunities in human genomics, with an emphasis on health applications. It is called the Forefront of Genomics, which contains the highest priority elements and vision for the cutting edge of human genomics. This is the 4-area strategic framework, which shows progressive and interrelated areas. Because of the growing complexities of human genomics and its many applications, especially in medicine, the consensus is that it is necessary to reaffirm, sharpen, and extend the following guiding principles and values in research. Focus on using genomics to improve human health, strive for global diversity in genomics research, maximize its use and access in healthcare, embrace interdisciplinary and team-oriented nature of genomics research, adhere to the highest requirements related to responsible data sharing and rigor in genomics research. A decade ago, we shared many of the challenges of developing countries as far as genomics research is concerned, such as the lack of coherent national policies, limited number of well-trained genomic scientists, poor research infrastructure, and local economic and cultural challenges. But things began to change with the establishment of the Philippine Genome Center in 2009, a purpose-built resource which seeks to expand significant collaborations, networking, and information exchanges within the Philippine scientific community from various institutions. This structure is a constant reminder for us to pursue academic and research excellence for the benefit of the nation. These are the general directions of the PGC: to accelerate the demand for NGS and high-throughput sequencing by crafting research proposals in collaboration with institutes in the different sectors, increase industry collaboration, and work on revenue targets for sustainability. Proactively work on legislation or national policies on sequencing Philippine samples, continue to partner with science champions for a better understanding of genomics and bioinformatics, and how S&T can contribute in enlivening the economy and increase the country's productivity. This is the roadmap for the health program R&D jointly developed by the PGC and the Department of Science and Technology. Our main objective was to apply genomics for the promotion of health of every Filipino through a Filipinized personalized medicine. We thought then that our short wins will be in the area of discovering biomarkers for susceptibility to communicable and non-communicable diseases as outlined here. But we also needed to be aligned to the National Unified Health Research Agenda that prioritized the top 10 causes of morbidity and mortality in the country. And so our R&D journey began with a health program utilizing genomics research to develop diagnostic kits for early diagnosis of diseases, strategic biogenomic surveillance, identification of new strains during pandemics, as well as pharmacogenomics. Funded projects from the Department of Science and Technology, Philippine Council for Health Research and Development included the development of the dengue kit, discovery of biomarkers for heart disease and diabetes, as well as for colorectal cancer. The lab in a mug device integrates and miniaturizes an isothermal unit as small as a mug, which functions as a portable laboratory developed by Dr. Raul Dystonia. For both cardiovascular disease and diabetes, we did multiple case control studies to test different hypotheses, response or no response to drugs, development of adverse drug events, and having the conditions of interest like complications. The results of our pharmacogenetic studies on cardiovascular diseases are seen on this table. The left column shows the phenotype for response to drugs and development of adverse events. And on the right are the genes where we found significant biomarkers that can be associated with poor response as well as side effects. Some of this work have already been published. Genetic association studies were also done for hypertension, dyslipidemia, stroke, and acute coronary syndrome. Shown here on the right are the genes where significant variants were found among our Filipino patients. For diabetes, the phenotype of interest was response or no response to commonly used anti-diabetic agents such as metformin, glycoside, and glimepiride. And on the right column are the genes with the significant SNPs, which can be used as biomarkers for response to drugs. We have equally interesting findings for our susceptibility to type 2 diabetes mellitus, which clinicians can use to predict the development of complications of diabetes or that leads to worse outcomes. In partnership with Dr. Ray Garcia's Disease Molecular Biology and Epigenetics Laboratory at the NIMBB, novel mutations were found among patients with colorectal cancer. These were functionally characterized and have been published in peer-reviewed journals. These provide evidence that interethnic variability exists, as tumor profiles of our Filipino patients are different from other population groups. And all of us have witnessed the unfolding story of the variants because we have a genomic biosurveillance in place. This would not have been possible if we did not have genomic sequencing and bioinformatics expertise at the PGC. These efforts have led to the detection of the entry of the variants of concern in the country. We were able to track SARS-CoV-2 transmission dynamics and even identify local signatures of evolution and emergence of new variants. The last part I would like to share with you is about the vision for precision medicine for Filipinos. This is not our vision for precision medicine. This is a picture of what we do not want to happen to our patients because we lack the knowledge and the tools to do so. This baby is an 11-month-old girl diagnosed with grand mal seizures and was started on an anticonvulsant to control her epilepsy. 2 weeks later, she had a severe mucosal blistering, life-threatening condition from a very preventable adverse drug event called Stevens-Johnson syndrome. Traditionally, the practice of medicine is a one-size-fits-all, and this is because our treatment guidelines look at disease uniformity. Patients are prescribed the same drug at the same dose, but we know very well that intervariability in drug response exists. And although a large majority of patients will probably benefit from that drug, some will not respond to the treatment, and others may experience very bad adverse events. In precision medicine, patients are stratified and can be given the right drug at the right dose the first time around. The best example is the anticonvulsant drug carbamazepine. It is the leading cause of Stevens-Johnson syndrome in Southeast Asian countries. The HLA-B *1502 allele has been reported to be associated with SJS among the Han Chinese, Thai, Malay, Vietnamese, and Indonesians. But this is not present among Koreans and Japanese. This association is also not present among people of European ancestry. A recent case control study by Dr. Capule and his co-authors from UP Manila investigated the association of HLA alleles with carbamazepine-induced SJS. They were able to demonstrate that HLA-B *1521 allele may be used as a genetic risk marker for Filipinos to prevent carbamazepine-induced SJS. Carbamazepine prescribing labels in other countries already contain information on serious dermatologic reactions associated with people who have the HLA-B *1502 allele. By including one's genetic information, treatment can be made more precise and personalized. It is almost like having your name written on the drug prescribed to you. As the definition suggests, the power of precision medicine lies in its ability to guide healthcare decisions toward the most effective treatment for a given patient, and thus improve care quality while reducing the need for unnecessary diagnostic testing and therapies. The components of precision medicine across the continuum of care are as follows: to predict disease before symptoms, for timely identification of cause, and for optimal treatment to be offered. These components reflect the move from focus on treatment only in healthcare to include prevention, prognosis, and post-disease survivorship. Implementation of precision medicine will require construction of a digital ecosystem which connects omics technologies to social, behavioral, and environmental factors. Curation of the dynamic data in the system, data analysis and reporting of predicted risk, differential diagnosis, and treatment optimization are necessary components of big data driving precision medicine. This is both a challenge and opportunity for genomics, bioinformatics, and data scientists in our country to collaborate to make precision medicine a reality sooner than later. This map shows the international endeavor for genomic medicine. Since 2013, the governments of at least 14 countries have invested over $4 billion in establishing national genomic medicine initiatives. All of these large-scale initiatives have the opportunity to transform healthcare systems by integrating genomic technologies into clinical care. There are two key priorities of this global effort to make this happen. First is evidence generation, and second is data sharing. Population-specific sequencing projects more accurately captures the natural diversity present in a population. It enables large-scale studies of specific ethnic groups to dramatically improve their representation in genomic databases. It can provide evidence for the clinical utility of genomic testing for precision medicine. However, the underrepresentation of non-white ethnic groups in scientific research and clinical trials has been a disturbing trend. human genomic databases are heavily skewed toward people of European descent. It limits how these databases can be applied to genomic research and to the clinical care of diverse populations. National initiatives, even in resource-limited settings, are needed and can often move more quickly in response to specific local health needs. We need to generate our own population-specific data. And fortunately for the Philippines, the Department of Science and Technology, Philippine Council for Health Research and Development has recognized this and has funded omics technologies for health projects that I have mentioned earlier, which are important in building the foundation for precision medicine. Through the Cardiovascular Genomics Program, a genomic database for heart disease specific to the Filipino population has been built. We also have a genomic database for diabetes. With the data that we now have, we will be able to offer to a certain degree the right drug at the right dose for the right Filipino. And this is in line with the Saktong Lunas para sa Saktong Pinoy initiative of the Department of Science and Technology. A cancer genomics program is also in place, and genetic testing services at the PGC is about to commence for hereditary cancer testing and hopefully soon also tumor profiling. A big part of this program is the development of companion diagnostic kits that are tailored for Filipinos. This is to make genomic testing available, affordable, and accessible to every Filipino. As I conclude this presentation, developing countries with limited resources like ours, we have the most to benefit from precision medicine, as we cannot waste precious resources from therapies that clearly do not work. We have to come together and come up with our own Filipino reference sequence to overcome a big barrier for precision medicine. This is a forthcoming project called the Filipino Genome Project, or the Filipino, an initiative to sequence 7,641 Filipino genomes representing the different islands of the Philippines. In this talk, I presented genomics at the forefront of improving human health, health genomics landscape in the Philippines, the opportunities that we have, the PGC health program's challenges, as well as future directions. Maraming salamat po muli at magandang hapon po sa inyong lahat.
Thank you very much, Dr. De La Paz, for that very illuminating talk on how genomics can be leveraged to address the health concerns of the Filipino people. I'm sure that our attendees are looking forward to hearing from our other invited resource speakers as well. But before we proceed with that, I want to note a few things for our audience. Please note that you can submit questions for all of our speakers in the Q&A tab during the presentations or at any time during the conference. I noticed that a couple of people already raised their hands during the keynote address. So if you have something you want to say or ask, you can find the Q&A tab at the bottom part of your screen if you're using the desktop view, or it's in the upper right corner of your screen if you're in mobile view. And then at the end of today's talks, we will be holding an open forum to give all of our speakers a chance to address any questions that you might have for any of them. And with that, allow me now to introduce our first speaker for today, Dr. Aloysius Domingo from the Center for Genomic Medicine and the Collaborative Center for X-linked Dystonia Parkinsonism, both at Massachusetts General Hospital. Dr. Domingo graduated from the PGH Adult Neurology Program in 2012. He is currently a postdoctoral MD PhD researcher at Massachusetts General Hospital, Department of Neurology, and the Center for Genomic Medicine, working with the Bragg and Telkowski labs to unravel the genetic architecture and transcriptional mechanisms surrounding X-linked dystonia Parkinsonism and other forms of genetic dystonia. He aims to build a research program interfacing cellular modeling and functional genomics to identify altered genes, networks, and pathways in relevant cell types in neurologic disorders, particularly dystonia, leading to the identification of potential therapeutic targets. Let us all now welcome Dr. Domingo, with his talk entitled Functional Genomics for Dystonia, Challenges and Opportunities.
Uh, okay, let me share my screen. Okay, do you see my slides?
Yes, we do, and we can hear you clearly, sir.
All right. So thanks for that introduction, and thanks also to the PGC for inviting me to give this talk today. As mentioned, I am Aloy Domingo, and I am a postdoctoral researcher at the Massachusetts General Hospital and Harvard Medical School. Today's talk will be about functional genomics in Dystonia, and I will be discussing the challenges and opportunities in this field. My research interest at MGH is regarding dystonia, which is a rare neurologic disorder characterized by action and co-contraction of muscles brought about by abnormal signaling in the brain. And functional genomics in dystonia is actually a very young field. It is because there are many challenges associated with the study of genomics in dystonia. In the first place, dystonia is a rare disorder, so it's not common to find people who you can sequence or biosamples from these individuals who manifest with a phenotype of dystonia. Aside from that, dystonia is also a neurologic disease and has a neurologic manifestation exclusively. So this means that any sort of modeling would have to involve neuronal cells, for example, either getting brains from mice or are modeling neuronal cells in cell culture, which is what we did. So that is a challenge that one has to overcome whenever dealing with neurologic diseases such as dystonia. Dystonia also has a varied mutational profile. There are missense mutations, there are loss of function mutations, and the genetic architecture is also complex, meaning that some genes code for many different phenotypes. Some of the different phenotypes converge also in single genes. And in many different instances, the penetrance is not even similar for many of the hereditary dystonic diseases. Some of them have 100% full penetrance, and some of them are modified by modifiers of disease expressivity and penetrance. So that is what makes this studying dystonia in a genomic context also very challenging. On the other hand, there are many opportunities brought about by advances in technology, especially in cellular modeling, as I mentioned, and also advances in our methods for bioinformatically analyzing sequences, and in particular, RNA sequencing. This allows us to create studies or use studies that explore the convergence of many different molecular mechanisms for the different hereditary dystonic diseases, but also for other diseases which has overlaps with dystonia, such as Parkinsonism and even neuropsychiatric diseases. So this is a very exciting time for this field, especially because we're starting to look at the translational potential of the molecular signatures that we have been looking at. And in today's talk, I'll be especially focusing on one very recent publication that we have in the American Journal of Human Genetics, wherein we looked at the challenges for studying functional genomics in dystonia, and then used RNA sequencing, cellular modeling, and bioinformatic methods to overcome those challenges. So missense and loss of function mutations in this gene called FAP1 cause isolated dystonia. In figure A, this is a figure of the gene body of this gene. This gene has a DNA binding domain, it has a call domain, and also a nuclear localization sequence. And human disease-specific variation has been described in all of those three different domains. Most of them converge in the DNA binding domain, which is the most important domain of this gene. Because we wanted to model disease-specific variation, what we did was to use CRISPR to introduce mutations in the background, basically in a stem cell background. So these different numbers and these inverted triangles show the different loci that we targeted. We targeted 4 mutations, N12K, S21T, P26R, and C54Y in the DNA binding domain. And then 1 mutation, which was benign, was targeted in the call domain. We also made a loss of function mutation in the nuclear localization sequence. And then we deleted the entire coding sequence of the gene using a dual guide approach. So 2 different guide RNAs at the start of the gene and also at the end of the gene were used to excise the entire coding region of this gene FAP1. And this we did because we wanted to look at different models based on known FAP1 biology. And we wanted to look at the functions of the different domains of different mutation types, such as loss of function versus missense and haploinsufficiency. And we also wanted to investigate what the signature would be of a benign mutation, such as M143V. So that's the challenge that we wanted to look at. We did the CRISPR in an isogenic stem cell background, immune pluripotent stem cell background. So these different experiments we did separately, and then we differentiated them into, as I mentioned, into neural stem cells, because as I mentioned, dystonia is a neurologic disorder. So one needs to model them in a neuronal cell type, such as an NSC. This picture in C just shows the immunohistochemistry that we performed, showing that we were able to indeed create neuronal cell types from immune pluripotent stem cells. And then we performed RNA sequencing. One of the things that we discovered using a DGE analysis, or differential gene expression analysis, was that the full gene deletion had the most profound transcriptome-wide effect. So in this bar plot here in A, you will see that there were about 2,500 differentially expressed genes between the deletion mutant and the control, whereas all of the other missense mutations and even the loss of function mutation R146 FS frameshift induced only hundreds of differentially expressed genes. But one interesting thing that we noted was that when we compared the mutational profiles of the missense mutations in the DNA binding domain and also the loss of function mutation in the nuclear localization sequence, we saw that these mutation profiles correlated significantly, as shown in this heat map in letter C. So there were common genes that were differentially expressed, whether the mutation was a missense or whether it was a loss of function mutation, as long as they belonged in the DNA binding domain or in the nuclear localization sequence of the gene. So different genes are shown here. GALC, for example, was a common gene, as well as this zinc finger, ZNF785, was differentially expressed in all of the different mutational models that we created. Because of this convergence in the mutational profiles, we thought that we could do a meta-analytic approach. And this meta-analytic approach identified a common set of 871 joint DEG genes that we termed as joint DEGs in our publication. And then we wanted to understand what these joint DEGs mean and what they functionally code for. So we performed gene ontology analysis, and this method called WGCNA, or weighted gene correlation network analysis, which looks at gene modules or groups of genes that work together under a certain pathway or functional annotation. You will see here that one of the associations that we found was a novel association for myelin-related terms, aside from identifying known functional associations with FAP1. So in bioinformatics, it's always important to recover known annotations. So here we identify the known annotations of FAP1, such as its participation in the P53 pathway, its participation in neuron development and in neurogenesis, also shown in this dot plot here on the left in A. But besides that, we also have identified a novel association. So this has not been described before, that FAP1 participates in myelination and in myelin-related processes. So this is something that we identified by using functional annotation of the joint DEGs that we identified using gene ontology analysis as well as weighted gene correlation network approaches. Because we wanted to validate our findings, so previously we were doing everything on NSCs and also using RNA sequencing. We wanted to validate our findings, first of all, in a different model. So we wanted to look at mice. We also wanted to validate it in different developmental time points. So remember that we were doing our experiments on neural stem cells, but here we're looking at mice which are just born recently and also in adult mice. What we did was we knocked in one of the mutations that we studied, which is the C54Y mutation in the DNA binding domain. We then also knocked out the gene, again using a deletion approach. And then we looked at qPCR and found age, region, and genotype-specific variations in gene expression in different brain regions of these mice that we mutated. So the three different regions are regions that have previously already been associated with dystonic disease, such as the cerebellum, the cortex, and the striatum. So all of those regions showed differences in gene expression in the mutants versus in controls. Besides that, we also wanted to look at structural integrity, so aside from gene expression. And so we performed Luxol fast blue stains of the corpus callosum. And in this picture in B, you can see that the myelination of mutants, this knock-in mutant and also this knockout mutant is not as prominent as compared to the wild type. So these are micrographs taken from the corpus callosum of dissected brains from these mice. So what did we learn from this experiment and from this study? We learned that we can use advanced technologies to overcome specific challenges in rare disease genomics. And those advanced technologies right now include using stem cell modeling, using genome engineering such as CRISPR, and my favorite, of course, using bioinformatics and transcriptome analysis to overcome these challenges. It also highlights that we can identify both common and novel disease signatures in many different kinds of mutations using functional genomic approaches in this disease. And once again, I would like to ask you to promote this publication that we have, which is just very recently published in the American Journal of Human Genetics. If you're interested in learning more about that study that we did, this is the title of our publication. And just to segue now into some local context, because we also have in the Philippines a very specific form of disease called X-linked dystonia Parkinsonism. This disease also manifests similarly to THAP1 dystonia. The difference is that this disease is endemic to the Philippines. Besides that, the clinical features are also somewhat different. Aside from dystonia, patients also manifest with Parkinsonism. So they start off with dystonia initially and then transition towards having combined dystonia and Parkinsonism, and later on full Parkinsonism towards the latter half of their disease. We discovered that this disorder is caused by SVA, retrotransposon insertion, in a different gene, which is TAF1. And hexameric repeats within this gene are also correlated with the age at onset of this disorder. What is the pathology? This disease is characterized by striatal degeneration, so degeneration of medium spiny neurons in the brain. And in 2018, we also published this article, and in this study, we dissected the mechanism of X-linked dystonia Parkinsonism, also using functional genomic approaches, and this is by integrating genome and transcriptome assembly. So similar to the THAP1 study, which we did more recently, in XDP, what we did was to take skin fibroblasts from patients, make induced pluripotent stem cells from them. And then similarly also, because this is a neurologic disorder, we differentiated these stem cells into neural stem cells into differentiated neurons and also a different kind of induced neurons. And then those are the models that we studied. I mentioned previously that this disease is caused, the genetic component of this disease is caused by an SVA insertion within the TAF1 gene. So we also used a dual guide approach to excise this insertion. So again, similar to how we did the deletion in the previous study. And that those delta SVA stem cells were also then differentiated into neural stem cells and finally into differentiated neurons. And then those are the models that we compared the XTP cells to. Using also RNA sequencing and bioinformatic methods and analysis, we found 3 different signatures for XDP. First, we found intron retention. So here in this picture, on the left, you can see that intron retention in red bars is identified only in XDP patients. It's not in controls, blue bars. And then when the excision is performed via CRISPR, this intron retention is reduced to the level of controls. Aberrant splicing is the second signature. So here again, we see no aberrant splicing in controls, aberrant splicing only seen in XDP. And then when CRISPR is performed, this splicing returns back to normal, similar to controls. Finally, TAF1 is downregulated in XDP patients. So red bars are here lower in comparison to control blue bars here. But when CRISPR is performed to excise the SVA, we increase back TAF1 expression to the level of controls. So this is our publication, and that's how we looked at and found those signatures for XTP. Finally, I want to say that all of this work was not done just by myself, but by a village, really, by a whole group of people, including people who collaborated with us from the Philippine General Hospital and the UP, from my previous laboratory in the University of Lübeck in Germany, and in my current laboratory at the Center for Genomic Medicine and the Department of Neurology and CCXDP at Harvard Medical School. I also want to especially thank my funding agencies, the Dystonia Medical Research Foundation, as well as the patients, participants, and families who helped us by giving away their blood samples and by cooperating with our studies. Please look through the different publications because this is just a 15-minute talk. And for any questions, please do contact me at this email. Thank you.
Thank you very much, Dr. Domingo, for that very detailed talk on the complex genetics and genomics of these neurological disorders. Just a quick reminder again for our audience, while we won't have a Q&A after each talk, we will have an open forum after all the speakers have had their turn. I noticed that some people already asked questions. Please continue to submit these questions. And to our speakers as well, feel free to answer the questions already. Type in your answers in the Q&A tab, in addition to responding to them at the end, whichever you prefer. And with that, I'd like to now introduce our second speaker. We have Dr. Martin Hibbard from the London School of Hygiene and Tropical Medicine. He is a professor of emerging infectious diseases. In addition to his position at the London School, he has adjunct positions at the University of the Philippines, Manila, as well as the Genome Institute of Singapore. He has a very broad scientific background, spanning both microbial and human determinants of infectious and inflammatory diseases. His current research interests utilize genomic applications to cover both pathogen and host aspects of infectious disease in order to understand transmission and identify novel therapeutic targets and approaches. His talk today, which will be about this broad topic, is titled Working Virtually to Understand More About the Virus. Let's all give a virtual hand to Dr. Martin Hibbard.
Thank you for the very kind introduction. Let me just share my screen, if I can. Hope you can see the screen.
Yes, we can.
Great, thank you. So I thought I'd take some time today to talk about not my usual work, but what's been happening during our time of COVID. So I think we've all been particularly busy during this time period, and struggling to keep up with our ordinary work. And this is some of the extra work that's kind of come our way during this time. And of course, I'm going to concentrate my efforts on working with the SARS-CoV-2 virus and the COVID-19 disease. And I think I'll start by just comparing if I can change slides, by thinking about this in relation to the original SARS back in 2003. So at that time, I was working at the Genome Institute of Singapore, and we were the first genome center to sequence multiple strains of SARS. And I think when we look now at how SARS-CoV-2 started, the initial part looked quite similar to SARS-CoV-1. But unfortunately, of course, it's gone in a different direction. And whereas in 2003, we were managed to control that virus outbreak and prevent it spreading too far, that hasn't happened with COVID-19. And I think now we're stuck with a new endemic disease, which we will all just have to deal with as best we can. And of course, the tools are different since then, and we've made huge progress. One thing is that while it took us a few months really to get to grips with SARS the first time around, the sequence for SARS-CoV-2 came out much faster. And that's enabled a worldwide effort to start the sequencing. We've heard a little bit about that earlier. I would say that the world has started doing the sharing process, and GISAID has been an important component of that. And I was very pleased to see that many of my colleagues that worked with me in Singapore at the Genome Institute there actually initiated this program, initially with thinking about influenza, but of course now it's moved on to SARS-CoV-2 as well as the original SARS-CoV-1. So the world effort is there, PGC is there doing all the genome sequencing, and in fact there is also a worldwide effort to try and understand those sequence strains. And this is analysis by next strain, which we've also heard hinting at. So in the London School, we do a lot of bioinformatic analysis of various virus genomes, and we thought what could we contribute in addition to these type of processes. So we thinking about what would be most important, and this is a little bit of an understanding of how the virus interacts with its human hosts. So the spike protein binds to the ACE2 receptor, and this binding component seems to be a very critical aspect to how the virus catches and then invades the cells. And variations in this here in the receptor binding motif highlighted in this 3D structure of the protein allows those binding systems, that connection to either to get more or less sticky. And we can see that the WHO listing of the variants starting alpha, beta, gamma, and others are differentiated really by those variations about how well they stick to that ACE2 receptor. And of course, now we have the Omicron variant, which we're still trying to understand exactly how those variations might influence this sticking process. So these mutations can be now identified, through the whole genome sequencing approaches, and we can then start to realize the impact of that. One of the revealing things is that this virus has generated these same mutations several times on the different backgrounds. So on these different variants, which have originated through different selection processes and end up with the same mutations. And so those tell us that not only are they very important, but we can understand them as they move from one to the other. And that applies to the new Omicron variant as well. But right at the start, we looked at this data in the London school, where I'm currently mostly based. And we thought, how can we interpret that in a slightly different way? And this publication in Genome Medicine came out a little while back now. And we were thinking about how the variants might be evading the immune system at some future date. Fortunately, that hasn't happened yet, but we need to be prepared. And this allows us to look within the genome and utilize our understanding of how it interacts, for example, with the T cell responses, and how well those epitopes of the virus, those virus components which are recognized by the immune system, can be predicted. And we put together a range of different tools which can look at that in some detail. And on the left, we're looking at the SARS-CoV-2 spike glycoprotein, the one that binds to the ACE2. And in dark green there is the receptor binding motif. And in the red dots across there are all the variations that were seen at that point. And what we're trying to do is to understand whether these would alter the recognition system, the B cell epitopes or the T cell epitopes. And on the right, we're looking here about the known binding, so the positions, the amino acids across the spike protein, and how they might be bound within a particular HLA alleles. Of course, we know that different ethnic groups tend to have a different selection of HLA alleles. And here we're looking to see which HLA allele and which binding position would have, and trying to predict how well the immune system might recognize the virus and its new variations. So far, we've been lucky. There hasn't been a proper escape mutant from this, but as I said, we're still looking at Omicron. Across the world we are. But there might be at some point in the future. There is some hope also, though, from the other coronaviruses that we commonly see, the common cold-causing coronaviruses, those which we've known for a very long time and which we frequently get each year. And on those viruses, they don't change that much. In fact, they're able to reinfect you each year because of a fading of the conventional immunity system. And that might be what happens with SARS-CoV-2, in which case you need those booster jabs, your third jab, and potentially a new top-up immunity jab every year, if that's the case. And then hopefully we won't need this analysis program to identify escape mutants. Not only are we trying to do analyses, but in this case, I'm working with my colleagues in Indonesia at the Eichmann Center there. And in this work, we've been trying to identify whether the diagnostics that we use for COVID are able to cope with a dengue cross-reaction. So I'm working with dengue around the world. That's part of my day job. And I'm certainly working with the Philippine Genome Center, trying to understand the diversity of dengue viruses across the Philippines. But in this study, we're trying to look to see whether that might cause trouble, either for the diagnostics. Of course, these diagnostics are developed in Europe and the United States, and they don't have the opportunity to look for cross-reactivity with dengue. And this study allowed us to look at that. So what we found is that a wide range of diagnostics, SARS-CoV-2, COVID diagnostics work pretty well in people with dengue. So there's not too much trouble in detecting it. But what we did identify is that there are some people who have possibly co-infected, dengue and COVID, and also those patients that are suspected of being COVID patients might actually be dengue. So you need to keep up with your conventional diagnostics and not assume that the symptoms that you're seeing are perfectly diagnostic. You need to do those tests. I want to talk now about work I was doing in Singapore, still with my colleagues at the Genome Institute of Singapore there, and working with the university hospital system as well. And in this case, we had the opportunity to do some transcriptomics, so RNA sequencing profiling, to see whether we can understand how the human host is responding to the COVID infection. And in this case, we were able to look at about 45 patients relatively early on, so 15 of them within 4 days of symptom onset, and 28 after 4 days, where we've got the PCR diagnostics and a diversity of ethnicities, and with a median age of about 35 in this group of infections. So we collected PAX gene tubes from these, and we did RNA extraction from the blood, and we did a differential express gene analyses. And because we got a relatively small number of patients, we were just a bit worried that this might be biased. So we did a data split, and we looked to see whether we could discover the same signature. And in fact, despite splitting up our patients in different ways, we were able to get a 67 gene signature, which distinguished active disease from those without disease. And when we look then by pathway analysis of the host response, you can see that with our KEGG pathway team, the top one was influenza A, but then we see a range of different other infections that come out from that. And on the pathways, we see programmed cell death, so that's typical virus infection process. But we also saw something that we hadn't seen in other virus infections. So these are the carcinogenesis, bladder cancer, acute myeloid leukemia, and a range of these cancer pathways which hadn't been observed. We looked from our other data sets, so this is work where I've looked at influenza, and you can see that there is quite a large number of genes that overlap between the COVID-19 signature and influenza. And across these genes, some of them give pretty similar expression profiles as seen in influenza, although some of them are a little bit higher in COVID-19. And there's a smaller subset which look similar to RSV, so respiratory syncytial virus, and overlapping with COVID-19. So we can see this inflammatory process overlapping with other well-known virus infections, and obviously that probably leads to some of the overlapping features, clinical features that we see. From these 69 genes, we were able to develop a risk score. So that's essentially just looking at the RNA-seq total expression of them, kind of adding up together all of those expression profiles. And we developed this risk score. So this is a number which is the relative expression in an individual person. And each dot here is a person with their RNA signature described by this single number, differentially expressed genes expressed by this single number. And we can see that this is proportional to the time from detection of COVID-19 disease symptoms. So our expression profile is related to that, and it's an early signature that we've detected. And by day 10, that expression profile has disappeared and probably changed into the next phase of the disease. And we can see that that also relates to the CT values. So what we're measuring is that, how much virus is in the samples in the nose, then our signature in the blood is expressing these signature genes at a higher level. And of course, the CT values and the stage of the disease are somewhat related to each other. So in the end, we can see that there is a median score. So this is our risk score, the transcriptome responses, and we can see that that relates quite nicely to the days from onset of COVID infection, and also from the amount of virus detected by PCR in the nasal swab. So this is a blood RNA transcriptome signature for COVID-19. And I think this is an early stage, initiates a strong host response, somewhat similar to other viruses such as influenza and RSV, but uniquely we identified cancer-related pathways. And this probably relates to a larger inflammatory response than we would normally expect. And future work could evaluate if this relates to disease severity. What we'd like to do is to identify early on in the disease, perhaps on symptom onset, who might be targeted for later treatment in the second week of the disease. And if we can do that, then we can use this type of risk score, which might be potentially transferred to a simple PCR test to help prioritize hospitalizations and who might benefit in particular from these, for example, antiviral therapies, which are now becoming available. So this is a little bit of a highlight of some of the extra work that's been coming my way during the COVID times. But of course, working in the London School and as a professor of emerging infectious diseases, this has been a lot of my work has been public engagement. I don't know, this on the top left is me in Channel News Asia, where I'm regularly asked to come in and talk about the European aspects of the disease. And in BBC World News, here I am early on in the process talking about our interpretation analysis, and more recently talking about the new variant, Omicron. I'm also asked to help with commentaries. So this is in the Lancet, a couple of articles there, and in the UK printed papers. So this is the Times and the Guardian and others. I'm also often giving my opinions, which are based on working with my colleagues across the world and trying to understand the COVID impact, from a genomics perspective. So with that, I'd like to end and thank you for the attention, and I'm happy to take questions about the progress and working in the time of COVID-19. So thank you very much.
Thank you very much, Dr. Hibbard, for a very comprehensive look at the different ways that you and your colleagues are addressing this pandemic, from looking at mutational dynamics to the test kits to host responses, all very important things to examine. And to our audience, as Dr. Hibbard said, he's welcome to respond to any questions. So please feel free once again to do so in the Q&A tab, and we will have an open forum after all the speakers have given their talks so that we can address those questions and concerns. And with that, we can take a short break for the meantime. We're actually slightly ahead of schedule, so we will take a 10-minute break. Let us resume at let's resume at 2:15. So this would be a great chance for everybody to stretch their legs or grab a snack or go to the restroom, grab some coffee, and we will be back at 2:15. Okay, thank you, everyone. Welcome back, everyone. I hope everyone's a little bit more refreshed. And we will now proceed with our next speaker, Dr. Brian Guevara from the Department of Dermatology of the Southern Philippines Medical Center, where he also completed his residency. To tell you a little bit about him, he is board certified in dermatology by the Philippine Board of Dermatology, as well as in dermatopathology by the Philippine, Asian, and International Boards of Dermatopathology. He has also served as editor-in-chief for the Journal of the Philippine Dermatological Society. And today, he is here to give a talk titled Molecular Dermatology, Path to Precision Medicine. It's my pleasure to introduce to you all Dr. Brian Guevara.
Hi, good afternoon, everyone. I'm Dr. Brian Guevara, Southern Philippines Medical Center. I'm a board-certified dermatologist and a dermatologist. My topic this afternoon is about molecular dermatology, which is the path to precision medicine. So what is molecular dermatology? It entails the study of genetics among the field of dermatology. This is an example of your gene where you will produce your hemoglobin and keratin, which will equate into your red blood cells and your skin cells. Very important in this history is that the Mendelian was, of course, established in the laws of genetics in 1865, and all those processes or sequencing methods by Sanger, and of course, the Human Genome Project, which is a decade of research and of course, expansion of our knowledge in genetics. So this from 90s to 2003, with the completion of the human genome sequencing, of course, this led to precision medicine. So single gene disorders counts around 7,000. And of course, there's going to be increase of new single gene or monogenetic disorders every year. So in the US, around more than 25 million patients, and this is, of course, accounts to almost $5 million because of the condition. And the Genodermatosis, which specifically is important in dermatology, there are around 300 entities involving more than 500 genes, and definitely affects the quality of life, not only the patient, but also the family as well. And there entails also psychosocial impact in the whole community. So these are the different examples of genodermatosis. You involve not only the skin, but of course the nails, the mucosa, the nail, the teeth, and so on. So more accurate diagnostics and better genotype-phenotype correlation are needed, especially if we manage these patients. So this is important in terms of establishing diagnosis, is that whole exome sequencing has produced a lot of entities that were discovered recently. So most mutations are located in the protein coding area, such as your exons. So exons are only 1 to 2% of your genome, such as in this image here. So it's quite accurate. And of course, it's going to be cheaper and faster in terms of analyzing all these mutations. So most are substitution, deletion, insertion, and there are also splice site variants in terms of the database. So in terms of identifying genes, so definitely you have to have the variant profile. So this is an example of affected individuals with your filtering and comparison. Especially we have to identify the association of an allelic series, and therefore after all these biostatistics accounted for, so you're going to have this positive variant in terms of your patient. So the cost, very important. It's very expensive when you had the genetic tests in the early 2000s. However, recently, at the end of 2014, because we have already 228,000 human genome sequence, and that's going to be really important, especially in terms of identifying all these gene mutations. So in terms of cost, definitely there's the decreasing trend in analyzing the data. For example, in 2019, around 30,000 pesos, that's going to be really cheaper compared to certain products such as your Apple iPhone. If you want to compare, this was like a few years ago, you have your iPhone X series or your iPhone 10. So it around probably 97,000. So now the latest iPhone 1 terabyte will be 100,000+. So that's going to be only 1/3 of the cause for your whole exome sequencing. So in terms of cases, I'm going to share you certain cases that I had encountered during my fellowship or training, and certain patients are really important so that we can understand not only the pathology, but also other applications as well. So this is an example of a patient with Blepharophymosis or small eyes, right? So you have small mouth as well, so micrognathia. So you have your fixed O mouth, you have your translucent skin, you have your prominent vessels as well. And because of the tight skin, there's going to be joint contracture. So there's going to be limited movement of the joints as well. So this is an example of a restrictive dermopathy. So this is the punch biopsy of our patient. So you can compare this to a normal skin later on. So you can see here that there's going to be flat dermal-epidermal junction. There's also thin dermal collagen fibers, and you have atrophic hair structures or adnexal structures. And in terms of special stain, BVG stain, you also lose the elastic fibers. If we compare that to a normal skin, so you can see here that you have very good epidermis here, you have your stratum corneum, you have also quite good collagen fibers also in the dermis. So we did the whole exome sequencing in this case, and also we found out that the father and the mother had both mutation of ZMPST24 gene. So if we do the histology, especially the immune staining, you have also the control here. So this is the protein ZMPST24 and laminin AC. So you have good signals here, okay, or you got good stains here in the epidermis. However, in our patient, there's loss of staining of the protein ZMPST24 and laminin. So this confirmed that the genetic mutation from the patient also had loss of proteins, which has to be translated supposedly. So what is the importance in the ZMPST24? So this is an example of a mouse model of progeria. So progeria, you have a very fast aging processes in an individual. So example of this patient with the ZMPST24 mutation, where you have this compared to the normal patient, or sorry, so a normal mouse, where you have this very good fur among these patients with the ZMPST24 in contrast to those without. So if you have those patients without the ZMPST24, similarly also with the mouse model, you will have alopecia or hair thinning or absence of hair. You have also hobbling gait, growth retardation, reduced grip and strength. You have rib fractures and death at 10 weeks versus that of a mouse, which has to be supposedly to survive up to 30 weeks. So this is a really good model with a mouse model of progeria. So let's understand further the importance of this protein. So the ZMPST24, which is a metallopeptidase. So it is responsible in terms of the production of your mature laminin A from your pre-laminin A. So in terms of laminin, it is important so that you will have a really good nuclear structure, as you can see here in the image below on your right side. So this is a normal nucleus with a mature laminin A. However, if you have a mutation or a defect in this metallopeptidase, you will have increased number of pre-laminin because it's not produced to laminin, and you have to understand that pre-laminin A is toxic, and therefore you will have defective laminin A, and you will have abnormal nucleus as well. So with that, you will have DNA damage, you have defective DNA repair, and you will accelerate your aging processes. So with the disruption of the nuclear architecture, with the blebbing and fragmentation, you will have increased chromatin conformational stress and genomic instability, which increases the production of your p53 and so your apoptosis, which is your cellular senescence. So that is why we have a common denominator among patients with restrictive dermopathy, your Hutchinson-Gilford progeria syndrome, and you have your Werner syndrome or progeria of the adult. If you can compare here, a 15-year-old female here at 48, you can see here almost 80 or 90 years old already where in fact she has to be at least 48 years old. So because of the accelerated aging, because of the disruption of the nuclear architecture, because of the gene mutation of the ZMPST24, therefore you will have this manifestation. And there entails also the potential treatment, because we have to understand that there is that meaning behind different mutations, and hopefully we can manage our patients in a precision medical way. So here, these are the different treatment options for our patients. So we have gene corrections, you have their splicing correction, you have also your protein correction, and eventually you can reverse cellular defects, and therefore you can have also cell replacement therapies. So technically, there's no other way in terms of reversing age, except if you are manipulating the laminin and your telomeres. All right, so to protect the DNA, you have to protect them with the architecture of your laminin. So we have understood that laminin is very important in terms of the stability of the nuclear membrane, so that you will have protective effects also from that of your UV, from the inflammation that is attacking your nucleus. However, also your telomere should be important as well, because this is a really important cap which will produce, of course, or avoid the senescence among individuals as well. So what is a telomeres? Imagine this as your battery. So for example, a patient with a longer or intact telomeres, you will have full bars. So these are, of course, your babies or your human adults or kids. However, whenever you have lower telomere and you have this low BAT already, so more or less this is an aging process or aged individual. So this is an example of a progeria patient. If you can YouTube this, Sam Burns, very important in terms of understanding life, because of course his philosophy for happy life is indeed from the heart, and of course from the experience of this patient. It's really remarkable. I want you to watch that as well in YouTube. Proceeding to the next case of this patient, this infant, of course, have their colloidal membrane. So it's like, or the patient is wrapped with this cellophane. You have your hyperkeratotic plaque, very scaly part of the skin, especially on the face. And you have your ectropion and your eclabium. So you can see here the eyelids, okay, or everted eyelids, and also your lips. And you have contractures and fissures as well. So these are really painful because of those fissures. So this is an example of a harlequin ichthyosis. Okay, so harlequin ichthyosis, in terms of the histology or biopsy, you can see hyperkeratotic stratum corneum. You have also preserved granular layer, acanthosis or thickness of the epidermis. And in the electron microscopy, because in Taiwan we had electron microscopy, you can see accumulation of the lamellar granules, which are abnormally shaped as well. So we did this whole exome sequencing as well, and we found out that there is indeed heterozygous mutation, specifically your ABCA12. So in terms of managing, these are your options for your patient. Of course, you have to admit the patient with under ICU. You have to correct the fluids. You have to provide nutrition, emollients, retinoids, antibiotics, and so on. Usually the mortality a few years back is around less than 20%. But now, because of the modern medicine, and we have understood already that the gene affecting the individual because of the keratinization process, there has been the use of a retinoid. So retinoids very important, so that you will have to normalize the desquamation of the skin. So these are the different preparations in terms of acitretin, and in terms of our case in a local setting, we have this at birth, at 10 months and 4 years, with marked improvement if you compare that at birth. So this was published in a local journal. So what is an ABCA12? It's actually an ATP binding cassette transporters. So this, imagine, is as a transporter which moves a certain molecule, for example, in this case, which is transporting a lipid outside the keratinocytes or skin cells. So that this lipid will lead to a barrier structure for the skin, and you have the normal desquamation process. So remember, we're like snakes, we keep on shedding every 30 days. So that's normal. If you have a defect in that transporter, you cannot desquamate, you cannot shed your skin, and that's why you have those hyperkeratotic or thick skin. So this is the normal desquamation process. And if you have this mutation, so you don't have the transporter, you cannot have that barrier. So you will have a defective skin and you will have accumulation of your skin as well. So as you can see here in our case, so without that lipids, without transporters, so you will have this phenotype, which is seen in a case. Okay. So imagine your ABC transporters as importers and exporters. So not only your ABC812, but you have a lot as well. So this is an example of a defect in terms of your ABC812. However, in your ABCC11, so that's quite interesting as well, because it can have the difference in terms of having a wet or dry earwax. So let's understand further your ABCC11. So it's like an oil rig. So it transports your oil or your lipid from the ceruminous gland to the surface of their ear canal. So if you have a normal structure of or function of your ABCC11, you will have a wet earwax. If you have a problem in terms of those, so of course you will have dry, but remember your SNPs, okay? So these are more or less normal variants. So you have your single nucleotide polymorphism. It is not necessarily abnormal, but it is quite distinct for a certain race or individual. So for example, a SNP, you have only a single nucleotide, for example, from C to T. So you have decreased function of that protein. So whenever you have decreased or altered function, you will have a dry earwax. So that's why there is a difference if you have a wet or dry earwax. If you have a dry earwax, you have that SNP. Okay, so if you can check your earwax later on at home, so you can identify whether or not you will have this kind of SNP. So if you compare a patient with a wet earwax, you will have well-developed or large lumina or your gland, and you have also your ABCC11 protein. In contrast, those with less developed lumina and also decreased protein in stain. So this is an example of a patient with a SNP. So we have with the normal wild ABCC11, which has a wet earwax. So definitely we have also different features, such as here, you will have body odor because of the glutamine conjugates. For example, your apocrine sweat glands. So apocrine sweat glands are usually activated during adolescence in the axilla. And of course, this will be responsible of that sweat odor, right? Because of the glutamine conjugates that excrete that certain substance, and the bacteria will of course have this reaction with the conjugate. Also in terms of treatment, so there's going to be drug resistance. So certain anti-cancer drugs are going to be pumped out on each cell. So it's going to be leading to drug resistance. And certain controversial studies also showing that there is decreased propensity of having breast cancer because there's low level of estrogen, and there's going to be a decreased risk because of that. So in terms of the different allele frequencies, so you can see here in African American versus that of the Asian components. So in African American, African and Latin American, so you have this ABCC11. And usually for patients with the SNP, such as the Korean, Chinese, Mongolian, so you have less likely to have a wet earwax and less body odor. So sometimes that's why whenever you ride in a plane, or probably probably a bus or what, probably you will smell certain odors because of the ABCC11. So because of the SNPs, that's why usually Koreans have dry earwax. They don't smell, even though they will have drug sensitivity and of course the breast cancer risk as well. So this is an example of my co-fellow. He's doing a PhD during that time when I'm doing my fellowship as well. So he doesn't take a bath probably just take a bath every 2 or 3 days. I asked him why, because he feels cold during that winter season. So he doesn't smell. And probably because of the SNP, he doesn't have that body odor. So I think that's an advantage also for that. So for us Filipinos, we have also certain SNPs, such as among Koreans, but not all are good in the sense that there's going to be breast cancer risk as well. So going to the next case, so this is a patient here in Davao. So you can see blisters in the whole body of the boy, also in the perioral area. You have also abnormality in the nail as well. So this is a severe case in Taiwan. When we saw this case, it is really in terms of heartbreaking, and you can feel that there is indeed excruciating pain. So the skin does not heal properly, and you will have fragility of the skin. So like the skin will blister because of touching probably or manipulating it lightly. So this is indeed at risk also of skin cancer. So this is an hereditary epidermolysis bullosa. So there are different classifications of EB. It's because of the abnormality in the dermal junction or the basement membrane, where you will have different classifications of the mutated protein. So what's good about the whole exome sequencing is that you will identify new identified genes every year. So similarly with the epidermolysis bullosa, just recently you will have also your KLH-L24. So this is quite unique because it is associated with your hypertrophic cardiomyopathy. Similarly, also with the CD151 just a few years back, this is also a new gene which is related to nephropathy as well. So in addition to those genes, of course, you will have a very specific or precise way of managing your case. So in terms of identifying EB, it's quite difficult. That's why we are proposing genetic testing among these patients, and that we propose in PCHRD and also your DOST. So hopefully in the near future, we can start our lab in terms of identifying epidermolysis bullosa in the Philippines, because it's so hard to diagnose because of the diversity of the clinical manifestation. And you have to classify it as major or minor EB because, of course, you have your different mode of transmission, phenotypic appearance, and of course, varied protein involvement that is important in terms of diagnosing these cases. So in terms of electron microscopy and immunofluorescence studies, these are important as well. But what is important in terms of understanding genetics of your EB is definitely the precise way of managing them, such as your genetic counseling, prenatal testing, and probably future clinical trial. And the more accurate diagnosis, the better genetic counseling and clinical care we can provide among these patients. And this is an example of algorithm or matrix. So we can download this for free in terms of identifying the specific type of EB. So we publish this matrix also in JPDS, in a local journal, the Journal of Philippine Dermatological Society. So basically, if you can see here your image among your patient, you just click on if it's absent or present, and it will provide a score like this, a diagnostic matrix score. For example, that is, for example, here, if you can save the report or print the report, you can see the highest score here is the EB-GS, or your generalized simplex. So it is around accurate to 95% of the time if you compare that to the genetic diagnosis. So this one, we propose that we have to do the matrix tool on the first part, and definitely we can classify this eventually with the help of this matrix tool. So an example of a case from Jennifer Tantatko, she's a pediatric dermatologist. So when I was visiting the Philippines during that time, so we were able to see this case, and you can see also blisters and erosions at birth. However, after 10 days with just wound dressing, there's a complete improvement also of the blisters and only leaving certain scars. And we, of course, tried to understand also what's happening in this condition. So we got the blood and brought it to Taiwan, where I had my fellowship. So we found out that there was indeed a collagen 7A1 mutation. And this one is very important because we are establishing a close ties with SPMC and Philippine Genome Center, and hopefully won't smuggle blood anymore to Taiwan. So this is a Taiwan case. It's like a deja vu. We were able to see a similar case also during those times. So there's a blister also at birth. However, at 2 months, there's also complete resolution, similarly to our Philippine case. So both also had a collagen 7A1 mutations. Both are underreported, and of course, no information in Google or PubMed. So we had to contact one of my mentors, Professor John McGrath in London or UK. So he is, of course, a gene hunter. So he has established certain collagen 7A1 cases, which he termed as bullous dermolysis of the newborn. So actually, he was able to publish this several years ago, 2005, and there was indeed a similar relationship or similar features from our case. Okay, so and why and how this correction occurs is not yet known. So this is your transient bullous dermolysis of a newborn by collagen 7A1. It is a special type of your epidermolysis bullosa, which is self-resolving. So in terms of patient care and support, there's this international group called DEBRA for people or for patients with EB or epidermolysis bullosa. Hopefully in the Philippines, we will have the same patient support as well, probably for the help of DEBRA or probably a national initiative from Philippine dermatologists as well. So again, it is not only genetics in terms of rare case or syndromes or obscure genes. It is also important that we will take care of these patients. That's why I was one of the group that proposed the study to DOST PCHRD, where we want to have a precise management for patients with EB and other genodermatosis in the Philippines. So we want to be a novel project that will cater genodermatosis all over the Philippines. During the pandemic, 2020, last year, and we were hoping that we will establish this laboratory already in SPMC, but because of the pandemic, sometimes there's going to be a delay in certain projects. However, it didn't stop us from receiving cases all over the Philippines. For example, in this first case, this is from Manila. So UP PGH Dermatology Department, it was referred to us. We have also similar EB cases like what you saw earlier and now also in around Mindanao. And this is a hairless gene, so a patient without hair. And we found out that this patient also had similar manifestations with the father and the older brother. And sadly, the patient will have risk of hepatoblastoma or liver cancer because of the gene. And hopefully we can catch that early in the near future. Another case, this fifth case, is from a Dr. Tita Barrio. She sent us images of the case and we were able to get the sample of the case, and eventually we were able to diagnose this as scleroderma pigmentosa, which is extreme sensitivity with sun, therefore producing a lot of skin cancer. All right, so if we can catch these early, so we can hopefully shift the course of the disease and hopefully to better prognosis for these patients. And that is why we have this collaborative plan with a group of your Philippine Dermatological Society, our society. We have also National Chiang Kung University in Taiwan and in King's College London, where Professor Macra is, and definitely the PGC, not only in Manila, but also definitely PGC Mindanao with Dr. Clark, of course. So what we have learned here about rare genes, of course, not only because of the rarity of the genes, but we can also have practical lessons from these cases. So for example, in restrictive dermopathy, we know how the aging process occurs. So if you were told by a certain influencer or a TikTok influencer that these are anti-aging creams or medicine, I think you should doubt, because of course you have to control the laminin and also the telomeres for you to have a true anti-aging regimen. Ichthyosis, in terms of identifying all the ABC transporters and exporters, you will have the different varied earwax and also your body odor. And in epidermolysis bullosa, there's going to be more and more cases eventually, not only in the Philippines, but worldwide as well. And with the help of the whole exome sequencing, we can have future, of course, direction in managing and diagnosing these cases. And these novel finds will, of course, need interdisciplinary research, and hopefully more and more of you researchers in the future would have this different studies, and particularly in molecular dermatology, because there is indeed a demand for new knowledge production, especially in the field of molecular dermatology, and there is still need to open with new possibilities, especially with clinical trials, and hopefully we can collaborate with that really soon. And I would like to acknowledge all my mentors in New York, UPenn, Yale, NCKU, McKay Memorial Hospital, and of course my institution in SPMC and Davao City. With that, thank you so much. And if you have questions, you can ask me after the lecture, and you can probably email me in my personal email. Thank you so much, and daghan salamat.
Thank you very much, Dr. Guevara, for that very eye-opening talk about these the genetics of these dermatological diseases and the effect they have on those patients. And also, thank you for sharing us what has been done and what can be done to address these diseases. And now, for our final speaker for today, we have Dr. Romulo de Castro from the University of San Agustin. He is a life sciences and health informatics professional and leader who assists organizations with biomedical research and data. He focuses on the transformation of scientific research and health information assets and data collection activities to create value. His recent work has involved various aspects of the COVID-19 pandemic response, including epidemiological and hospital resources modeling, contact tracing, PPE needs, vaccines, and genomic surveillance visualizations, which will be the topic of his talk today. Other pursuits include health data interoperability and data governance, mental health information systems, health mapping, telehealth, and community monitoring of marine protected areas. He also served as the director of the Center for Informatics at the University of San Agustin in Iloilo City. His talk today, which will surely be of interest to many people here, is titled COVID Genome Biosurveillance Visualizations in the Philippines and Southeast Asia. Can we assess Omicron using immune informatics? Dr. Jong de Castro, the floor is yours.
Many thanks. Let me turn on my video. Hi, oops, sorry, there you go. It's evening here. I'm in California, but I'll be back in the Philippines soon. So I'm looking forward to seeing you all, particularly Cynthia. Hi, Cynthia. And also Eva. Thank you so much. Inspiring talks from both of you and all the other speakers. So I guess what I'll do now is share my video. Correct? Tama ba? Ayun. Share my slides is what I mean. Okay. So let's see. Can you see my slides now? I'm gonna have to. Let's see. Here you go. Does that show up?
Yes,
Okay,
we can see it.
Great. Many thanks, PGC, and good afternoon to everyone. Good afternoon to all the attendees of this wonderful Genomics and Bioinformatics conference. Thank you for inviting me, and I'm really happy to give this talk because I'll present the things that the researchers and the students at the Center for Informatics have been working on for the last few months. A lot of them have been involved in the COVID-19 pandemic response from the very beginning. They've done hospital resources modeling, epidemiological modeling, PPE needs, contact tracing, and we even have a machine learning and artificial intelligence group, and we've done some work on classifying lung X-ray images, whether or not they are COVID or non-COVID. And we joined an international competition for that, like these inexperienced computational people. And, wonderfully, the very first time we joined the competition, they got a bronze medal. So I'm very happy for that. So I'm Romulo de Castro, Director of the Center for Informatics, and I will present our visualizations on genome biosurveillance. And also, maybe the one that you might be interested in, is our preliminary work on assessing whether or not, some of these variants, are escaping vaccine function. So, all right. So let me first. Okay. This is the project team. Mainly, this is the work of Jonathan Rico and Dominic Bolinas. Jonathan has an MS in physics, so he's a completely computational fellow. And Dominic is an MD PhD student at UP Manila. We're very proud of him because he also did some analysis of the data, the Philippine data, and found some interesting things which we reported to the DOH very early on. Jonathan has been involved in so many things, and from program officer of the DOST, he came back to the sciences, and now he programs bioinformatics tools. He visualizes genomic data, and does a lot of machine learning and artificial intelligence stuff. Also Jimuel, who works for Mr. Jimuel Celeste, who works for PGC, also contributed to this work, and also Nelo Aguila, Julius Villagracia, Pia, an MD PhD fellow, and also a high school student who helps us regularly, Isabel Isip. We've consulted a number of our collaborators from locally and abroad: Manny Bacolod, Badong Kawili, Bern Musman, Armando Solis, Doug Fenger, and they've been very generous with their scientific advice to us. So, all right. So let me proceed then. First, let me show you our visualizations of COVID in the Philippines. So we've been following COVID since the very beginning, from April 2020 all the way to now. And you can see, our ramp up to testing. The yellow graph is actually the number of people being tested, and the red is the number of people who turned positive. So you can see, a ramp up to our testing capacity here, and we kept going up. But you can also see, our some of the surges that have happened, and highly suspecting that these may be because of some of the variants that have entered the Philippines. But then we had vaccines starting April 2021, which brought down the cases, we think. And then we had another surge, which is very likely because of the Delta variant. And then, of course, we vaccinated more of our population. And now we see that there is a big difference with more vaccinations. So now the positivity rate is the lowest I've seen ever since the beginning. So it's at 1.7%, and hoping that that would continue. But now we have Omicron, so it remains to be seen, what will happen to our positivity rate, going beyond today. But we've also seen that at the highest point we have like almost 100,000 tests daily. But somehow some of that testing has gone down. So we don't know. Some of the things that we see here, are not very easy to explain, but that's what the data is. So we'll just keep following it, as we go. So hopefully, we'll get to better trends, in the future. All right. So we have these culprits of the surges, and the most associated is the Delta variant, which is has been taking over the world. We've had Beta, Alpha, also Gamma, and now Omicron, which was first detected in South Africa. If we look at the trends over time, this is how it looks like. And, clearly Delta is still, the most prevalent variant in the world, particularly in the US. But, look at Omicron. So it's coming up quickly. And again, we'll have to see what happens with Omicron, and everybody's crossing their fingers that, we have better trends. This is just an overview of the genomic surveillance that we visualized for Southeast Asia. And the reason we did Southeast Asia is because, of course, we want to know what's happening with our neighbors, because, although there's restricted travel across the region, we still want to know what might, enter, come in and out once we open to, our country to tourism, particularly regionally. So you can see here, we plotted the variants that have been observed in the region by country. So in the Philippines, you can observe, Alpha, Beta, Delta, and others. And actually, all over Asia, there's the same trend, except maybe for Gamma. So we don't see much of Gamma in the region. So it probably didn't get to our region, and that's well and good. So this is our overview, and you can see, of course, that in the different countries, pretty much a lot of them have been taken over by the Delta variant, in particular Singapore and Indonesia. So most of the viruses that are being sequenced in these countries are already the Delta variant. In the Philippines, we still have, or we've, we've had, we still have a large number of beta and alpha. So if you look at the the genomes of viruses of the variants over time across across Southeast Asia, you can see that Delta is still really the dominant variant, and essentially has taken over the the other variants. In the Philippines, we still have beta and alpha and beta, but we also are starting to show a lot of Delta. Unfortunately, the Philippines, there's a lack of data from, I think, August onward. So this is really old data from the Philippines that was, which we have. And the same thing for Myanmar, there's a data gap, and also Vietnam. But the rest of the other countries in Southeast Asia are, are updated up to November in their submissions of genomic sequences of variants. The source of all the data that we've been visualizing is GIS8. This was mentioned earlier by Dr. Hibbard's presentation. So GIS8 was originally for influenza, but now, ever since the pandemic, it's been the repository for molecular, for genomic sequence data for COVID as well. So to date, there are, not, 5.6 million genomic sequences in GIS8 from all over the world. And the World Health Organization encourages countries to submit weekly to GIS8, the sequence, the genomic sequences that sequencing that each country is doing. So we looked at how well we are doing in terms of genomic sequencing with respect to the other countries in Southeast Asia. And the good news is that we are actually by far submitting the most number of sequences to GIS8. So congratulations to PGC, probably one of 4 institutions that have been submitting genomic sequences. I think the other 3 are RITM, UP, NIH in Manila, and also the armed forces. I think it was, I don't remember the initials now, but I think it might, it's another a facility that's associated with the armed forces. But correct me if I'm wrong. So this is good. We're submitting a lot of sequences to GIS8. However, if you compare how frequently we make those submissions, we're not doing very well. We are, on average, the Philippines makes submissions every 19 days. And in terms of, this metric called collection submission time, we are actually, we have a difference of, our figure is about 3 months, so about 120 days between collection and submission. So, and that lags behind a number of the countries in Southeast Asia. So good that we have a lot of submissions, but we're not submitting as frequently and as fast. But in fact, all over the world, this collection submission time lag is actually a problem. And you can see here, the Philippines is at 50th place out of 54, with UK as the top, with the smallest CS collection submission time lag. Of course, naturally, that would be the UK with their sequencing ability and the excellent facilities over there. So we have a lot of catching up to do. Even the US is not necessarily doing very well. So, but you can see that, there is this disparity that has been pointed out in terms of sequencing. And, we can't have that. Disparities are never good, in health concerns. So we looked at, the regional picture in the Philippines. because there's that data that was associated, with that collection site data that's associated with the regions in the Philippines. So we have actually a good picture of, the variants that are present in the different regions in the Philippines. So you can see most of the sequencing still occurs from samples in NCR, followed by region 4A, and so on. And if you look at the submissions to GISAID, it looks very nice, nicely following, I meant if you look at the number of individuals that are positive. I think these are the number of samples that turn positive from different regions. And you compare them to the number of samples being sequenced per region. It looks very nice, a nice, good correlation with the number of positive individuals in the different regions. However, if you look at the percentage of the cases that are being sequenced over the number of cases, then you can see that there's not a very good visual correlation, where in NC in most of the country, only 0.5% of the samples are actually being sequenced, and there's a bit of a disparity across, where region 2 is doing very well. But NCR, of course, lags behind the number of samples being sequenced, which is strange because NCR is where all of those sequencing facilities are located, if I'm not mistaken. So this is this has to do with representative sampling over the region. So we're not seeing a true picture of what's happening in terms of the variance in the regions, because we are not sampling representatively. So we are sequencing a lot of samples, but our sampling methodology needs a little bit of improvement. So that's what what we are seeing in this picture. So we do have to get our efforts at representative sampling better. Okay, so now I'll jump into the other aspect of my talk, which is looking at the Omicron variant and asking the question: are the current vaccines going to be effective against Omicron? So we just put new variants in general because we developed a tool that should be able to answer the same question for each variant that shows up. And this is very preliminary work, so but we thought it was fun to show it just so we can elicit some feedback. Okay, first we are looking from the point of view of immune epitopes and vaccines. So an epitope or an immune epitope are like unique identifiers in the virus that the immune system recognizes either through natural infection or with the help of vaccines. So these immune epitopes are actually what activates the immune system against the virus. These immune epitopes are actually scattered all over the proteome of the virus. And this is the proteome, and you can see the proteins that comprise the virus, the most important of which is the S protein or the spike protein. Our vaccines are made differently. So for example, the Chinese vaccines, Sinovac and Sinopharm, are inactivated whole virus vaccines, whereas the vaccines from AstraZeneca, Janssen, which are adenovirus vector vaccines, as well as the Pfizer and the Moderna, which are the mRNA vaccines, are all developed out of the spike protein. There's the Sputnik vaccine, which has been distributed in the Philippines from Russia, is also an adenovirus vaccine out of the with the spike protein component inserted into the viral vector. And the component virus that was used for all of these vaccines are still actually the Wuhan, the original Wuhan strain. There's talk now of re-engineering these viruses to be able to address the variants by incorporating, the newer variants into the vaccine. So, but as of now, all of our vaccines are still based on the Wuhan strain. So there was a study early on, actually from my own group in La Jolla, that looked into the immune epitopes of COVID because they had sequence similarity to the epitopes that are well known from the original SARS virus. So for the S protein, there are about 28 epitopes that are B cell epitopes as well as T cell epitopes. And T cell epitopes are further divided into two types, class 1 and class 2. So for T cell epitopes, they have, as Dr. Hibbard said earlier, they have a particular restriction, which is which means that the immune activation occurs only when this restriction occurs. So these epitopes, of course, are of global importance because you can find all of these HLA across all over the world. But in specific races, they only have a subset of HLA. So that the epitopes that are relevant to a particular group of people has to do with what HLA molecules that population has. So vaccine efficiency across races is not necessarily similar simply because the immune epitopes that are responsible for immune activation against the vaccine and against the virus, still has a restriction, and that causes some sort of like subcompartmentalization into populations of the different immune responses. All right, so this is our epitope scanning tool, which was developed by Jonathan with the help of Dominic. So first, you can, we laid out the epitope types. So B cell, T cell class 1, and T cell class 2. This is the reference strain, which is the original. So this is for the spike protein of the original Wuhan sequence, Wuhan strain. So you can see it here. These are the test, these are the sequences that have been submitted to GISAID that we picked up. So these are the Omicron sequences. The moment, I think there's about 200 of these sequences in GISAID, Omicron sequences, or maybe there's more now, because just earlier this afternoon, Jonathan told me that he was looking at GISAID and he found submissions of Omicron from Singapore, I think Malaysia, and some other Asian countries. I don't know if we already have that in the Philippines, but maybe PGC can let us know if they've already submitted Omicron sequences to GISAID. But it was only it was like several days ago that we did this analysis, so there were no Omicron sequences yet from Southeast Asia when we when we downloaded these sequences. So basically, here is where your class 2 epitope is, which is a small segment of the protein. And if you scroll through, well, this is just a slide, so but our actual tool is that's live. You can scroll through. But basically, what it does is it scrolls through the entire spike protein sequence, and then you would then be able to see where these epitopes are located, and then you can see these mutations. All right. So so for example, this mutation is from S serine to I think P is proline. We labeled it as green as a conservative epitope, and I'll tell you about what a conservative epitope is later. And on the other hand, this particular mutation is from S, again, serine, to F, phenylalanine. This is a radical mutation, and we color-coded it as red. So in this particular epitope, which was hit by 3 mutations, there's 1 conservative mutation, and there are 2 radical mutations. So, and then if you scroll along, the sequence of the entire spike protein, you can see, you know, the mutations across the different epitopes, those 28 epitopes that I mentioned. Over all of the, you know, over all of the sequences, Omicron sequences that have been submitted to GIS8. So here, you know, this class, again, as a summary, this class 2 epitope was hit by one conservative mutation in 20 out of 26 samples that were submitted. So this is about 26 samples. And 2 radical mutations in 19 out of 26 samples. So I guess here, you know, there's, this is like, it retained the same, blank means it retained the same residue. So the assignment of whether mutation is conservative or radical was through the Grantham difference in matrix. And what it is essentially assigning a score, which is calculated by this distance formula, you know, to the substitution. This distance formula is contributed to by composition, by three variables, namely composition, polarity, and molecular volume. And they're corrected by the scaling constant. So basically, when you have a substitution, so these are like the amino acid residues, and then, you know, what they're substituted with. And so, of course, A to A is, there's no substitution. So diagonally, this is all 0. However, C to A substitution has a score of 195, which is calculated out of this distance formula. And the Grantham index, you know, gives all these numbers, but you know, this reference back in 1985 assigned that any number greater than 100 is radical, and of course 100 and below would be conservative. So this is what we were following. All right. So here it is. These are the 28 epitopes. And again, you look at the conservative mutations and the number of hits here. And you can see, and this is the number of the samples that are affected out of 200. So for this particular epitope, a B-cell epitope was only hit in 1 out of 200 sequences that we have. So that will change, of course, when we have more Omicron sequences. So this is about 0.5%. So you go through all of those epitopes and you can see all the conservative mutations, but not across all samples that have been submitted. The radical mutations also you can evaluate. And you know, there's a couple of interesting observations. So first, for this particular epitope, epitope 18, which is a class 2 epitope, there are, let's see, there were 2 radical mutations that occurred in 80% of the samples. And in epitope 24, there was 1 radical mutation that occurred in almost all of the samples. And if you have to assign a physiological function to these epitopes, so B cell, T cell, class 1 and class 2, those functions would have to do with the transmissibility. Class 1 epitope would be the disease severity, and class 2, T cell class 2 would be both transmissibility and disease severity. Of course, transmissibility is not necessarily just a function of B cell epitope immune activation. What I'm saying is that immune activation through B cell epitopes contributes to transmissibility, and the same way with disease severity. So not, I mean, immune activation, of course, you know, is probably a good component of it, but it's not all. So, but these were the only ones that we were evaluating. So in summary, we only saw 2 class 2 T cell epitopes affected by radical mutations. And the implication of this is, if you have a radical mutation, it is entirely possible that the epitopes may not be recognized by the immune system in those who have been vaccinated with the Wuhan spike protein. So if you think about it, the remaining 26 epitopes would still be recognized. And this 26 epitopes, all of these 28 epitopes are distributed across the population, across the global population, not in one particular person. So the more relevant denominator for a particular person is actually less than 28. However, if you are considering the 28 epitopes, then the more appropriate question would be whether or not the vaccines globally, the vaccines that have been rolled out globally, might have a global effect on, might still be effective globally on the Omicron variant. All right. So it looks like, at least I think, by this particular evaluation, we might actually do well against the Omicron variant, because most of the epitopes remain unaffected, but of course it remains to be seen. Now, laboratories, and including the vaccine developers, are evaluating with laboratory methodologies whether or not the vaccines will still be effective on Omicron. So they're looking at B-cell antibody neutralization assays, as well as T-cell cytotoxicity assays to evaluate the function of the immune system against the Omicron variants in vaccinated individuals. So it remains to be seen whether this rough prediction of ours will hold. And again, there might still be other refinements that we can do to this epitope scanning tool, including also refining the assignment of radical and conservative mutations with other indices that are also used in structural biology to describe protein function with respect to amino acid changes. All right. So in summary, there have been challenges in the emerging stronger variants, and Omicron is not the last, I promise you. And all of them threaten vaccine efficacy. And we've seen, sequencing disparities across countries and even across regions within the Philippines. And, with respect to the large lag in collection and submission time. So there's still a lot of things that could be improved on. However, there are also opportunities in developing some tools to do quick assessments on vaccine efficacy, such as this one, and also improving genomic surveillance and monitoring by, again, improving, sequencing, genomic sequencing representation, as well as processing of those sequencing data. And lastly, all of this, of course, will lead to more data-driven policies that may have an effect on, how we manage our health protocols on a national scale or even a regional scale. So the question is, if you were asking questions about, should we lock down again or not? And everybody is locking down actually in various parts of Europe, as well as, in the United States. I don't know. But maybe I'm a little nervous because I'm planning a trip back to the Philippines next week. So we will see. But we should have. I mean, I guess maybe the message is maybe let's not panic quickly, and just make educated and scientific assessments whether or not our tools against these variants are still effective or not. And of course, in the future, we will have many different, we're looking at many different variants and many different vaccines targeted to these variants. It may give us a different picture of our surveillance, wherein, we'll have, we'll know what variants are being observed in the different regions of the Philippines or the different regions of the world. And when we have variant targeted vaccines, then we would know what vaccines to roll out in the different regions, just if we just know what variants are occurring in various parts. All right, that's it. I hope you enjoyed our work at the Center for Informatics. We continue to work on COVID, particularly in data analysis and representations of the data, and other tools such as machine learning and bioinformatics. So thank you so much for the invitation, and well, you can ask me questions if you like. Thank you.
Thank you very much, Dr. De Castro, for that very engaging talk. I can personally attest that the biosurveillance team at PGC took great interest in your discussion, as your insights about visualizations is directly relevant to our work. And I like to think that your discussion on the epitope mutations may address some of the fears or concerns that a lot of people have about this emerging Omicron variant. And with that, it is now time for our open forum or Q&A for to close out this the day's events. So we will soon be opening the floor to the speakers to address whatever questions our participants have submitted. And once more, I'd like to call upon our everyone listening to submit questions to all of our speakers regarding any aspect that might interest you or concern you. We still have plenty of time and space for questions. That being said, I'd like to call upon our speakers now to be ready to turn on their cameras and microphones to answer the question. And I think the first, we can start off our Q&A with many of the questions that were submitted are all relating, circling around the particular topic of precision medicine. No? So let me read them all together. So the first one asks, when will genomics be integrated in the clinical setting, in which doctors would know what to test and tests would be both affordable and accessible for Filipinos? What are the current challenges and potential solutions in fulfilling this? Another person asks, how accessible or affordable might precision medicine be to the Filipino masses? Would these be covered by PhilHealth or other social funding programs? Another asks, how much do you think is the fair price for precision therapy? And yet another asks, when and how does the panel think precision medicine might take off for common use in the Philippines? So I think there's a common thread here of people wondering about accessibility, affordability, I guess, inevitability of precision medicine in the Philippines. So I invite all of our speakers, especially I think Dr. Cochonco de la Paz, Dr. Domingo, Dr. Guevara might be the ones most equipped to answer this, but of course you're all welcome to chime in. Would any of you like to say a few words about emerging precision medicine in the Philippines? Yes, Dr. Cochonco de la Paz.
Well, thank you. First, let me congratulate the speakers who gave excellent talks. Thank you, Aloy, Martin, Brian, and John. Thank you for enriching our learning experience on this day one of the Genomics Bioinformatics Seminar on Health. Now, thank you for the questions that were read by Carlo. As a geneticist, we have been using genetic tests for many years. I've been practicing genetics now for more than 20 years in the country. We use it for confirming a genetic disease or susceptibility for a genetic disease that runs in the family. So for many years now, we have been sending out samples to certified, what we call CLIA, certified laboratories abroad, like Invitae, Myriad. Of course, the cost has always been an issue. And that is why the Philippine Genome Center has established its clinical genomic services that will leverage the center's expertise in sequencing technology to provide molecular diagnostic services for clinicians and their patients. We have done some prioritization, and as far as the tests are concerned, we will offer in the beginning what we think is most important, and that would be on hereditary cancer testing. Of course, I know that diseases like rare diseases that such as what Dr. Brian mentioned in his practice in dermatology also is of prime importance. We also give importance to rare diseases, especially our team at the Institute of Human Genetics, because we do take care of a large proportion of these patients who need We're giving them enzyme replacement therapy, and we are, of course, we do a lot of molecular testing for confirming their diagnosis. Now, just to mention that our greatest challenge is really the cost of reagents, which makes it difficult for us to compete with commercial laboratories, even if they are based abroad, no? Because right now, I do send a lot of samples to a commercial lab in California. It's called Invitae. The cost of the test, whether you do a single gene test or a panel of genes, it only costs $250. We add on the cost of doing the transport, packaging, and of course, all the BOQ requirements. And I'm talking about, this is not for research, this is for service testing, no? The total cost is going to be about $304. How can you compete with just 17,250 pesos? But don't worry. As I said, we're doing our best at the Philippine Genome Center to make the tests affordable to the Filipinos, and we don't have to send out samples. We also need to recognize that human genomic databases, as I mentioned in my presentation, they're heavily skewed toward people of European descent. So it limits how these databases can be applied for genomics research and to do clinical care for diverse populations. But having said that, opportunities also exist, including development of companion diagnostic kits that are tailored for Filipinos, which is exactly what we want to do at the Philippine Genome Center. One of our greatest responsibilities for precision medicine to happen in the country is to make genomic tests accessible and affordable to every Filipino wherever they are. So the expansion of the PGC facilities to Visayas and Mindanao, this empowers us to do just that. So for PhilHealth coverage, there was a question about PhilHealth coverage. Right now, the NICA law, the National Integrated Cancer Control Act, in its implementing rules and regulation, it includes coverage for genetic testing as part of the cancer care. That's all I want to say. I hope I have addressed the questions that were raised earlier as far as precision medicine is concerned in the Philippines. Thank you.
Yes, thank you very much. Would anyone else like to chime in with some additional insights about precision medicine, particularly in the Philippines? Yes, please go ahead, Dr. Domingo.
I think I just want to reiterate and reemphasize what Eva said, which is that the first step really is to establish the infrastructure, including the Philippine reference genome. I think that that's a very important first step. We will, although there's Nomad and all of these other databases, they're heavily skewed towards Caucasian genomes. And it is important for us to establish our own reference sequence so that we can interpret variation correctly. Part of this is, of course, creating the task force in the Philippines, but also getting specialists to work on these genomes and to move forward with establishing that resource. This is a resource that we need to start now, because otherwise we will not be able to move forward with precision medicine until we take this first fundamental step.
Thank you very much. Those are very appreciated words. I believe Dr. Hibbard has something to say.
Yes, I would just add, it's excellent description, and of course all of that is needed. But precision medicine is actually a wide, broad concept, and including, for example, TB sequencing, which I think might be introduced across the Philippines sometime sooner, perhaps. And this is something which is affordable and has been shown to be cost effective. And it's something which I know the team is working hard to introduce across the Philippines. So I think this would be affordable and a good replacement. So yes, precision medicine is coming. It's already here, and it's just a matter of expanding. So thank you.
Okay, yes. Dr. De Guevara, am I correct that you wanted to say something?
Yes,
Yes,
yeah.
go ahead, please.
Right. Also, the definition of precision medicine, it entails not only diagnostics, but definitely it's into therapeutics as well. I understand it's going to be really expensive if we all base it from genetics. But what we want supposedly to have it more accessible and affordable are biomarkers. For example, some such cheaper alternative for precision medicine or diagnostics or genetic diagnostics will be, of course, immunostaining, some marker biomarkers that you can get only from the blood that could be really quite accessible for our patients, and it won't really necessitate the big laboratories that we usually send it abroad. And one of the first steps, for example, for managing psoriasis is quite common in the Philippines. 2 to 3% of the population of the Philippines will have psoriasis. And certain patients with pustular psoriasis or this subtype will have interleukin 36 RN mutation. And not all typical drugs for psoriasis can help manage that kind of subset. So what we are doing now is a clinical trial that targets interleukin 36 pathway. And with that, I think that's an option for patients in the future that we can treat them specifically on their phenotype. So we can more or less manage our patients because of the phenotype, because of their presentation, and hopefully we can manage them without the test, for example, for genetics and so on. For certain rare diseases, I'm really passionate about the rare diseases in dermatology. So we have a few. I saw a question there: how can we manage patients, or we do research whenever we have small sample size? So what we usually do in dermatology or in our institution is to collaborate. So we have a national referral center that will be responsible in collecting all these rare referrals. So for example, I'm sure Dr. Eva has a lot of rare referrals, rare genodermatosis probably, that we can have to collaborate so that we can manage these patients. And hopefully with our future proposal for PCHRD or DOST, we can set a lab that will receive all these rare genetic conditions, and not only in dermatology, but also in other fields as well. And hopefully we can manage our patients. In precision medicine, it's not only the medicine that we entail to discover, but also the functionality of these patients. Functional medicine is another aspect in medicine that we have to understand, and we have to modify, for example, our nutrition and so on. That's why in dermatology, we have also functional dermatology. Finally, in cost, of course, admittedly, it's going to be expensive as of now, but you can see in my lecture, there's a decreasing trend of doing genetic tests. Since early 2000, it's going to be really expensive, but now I think it's a cost or less than the newest iPhone. So I think that's a really good potential in the future, and hopefully we can manage our patients with the help of genetics. And with that, I think we have this opportunity, this discussion. I'm really happy that people or our audience are asking about precision medicine, because ultimately, from evidence-based medicine to precision medicine, I think that's the ultimate goal for our patients.
Thank you very much for that. And I'm also glad you touched on the question of small sample sizes. But before, we can look back to that later. But I think Dr. We'll close the discussion on precision medicine with some last words from Dr. Cochonco de la Paz.
I just wanted to mention, and I will ask Aloy to share his thoughts about this. He mentioned about XDP, a condition that is a uniquely Filipino disease, right? I think he has demonstrated in his presentation. Dr. Jong has to come into this. I think you have to pin Dr. Jong. He's not in the panel yet. I can see.
Noted, ma'am. Thank you.
Okay, so sorry. Cynthia and I love Dr. Jong. Okay, and of course also Martin and Aloy and Ryan. Well, I wanted to say Dr. Aloy mentioned this about XDP, how it's very important to understand the underlying biology. And what Aloy, what I want you to share is about how understanding the underlying biology of XDP is now allowing us to do, because we collaborate, no? Aloy and I were part of the Collaborative Center for XDP at the Massachusetts General Hospital in Harvard. Aloy, can you share the work that's being done on repurposing of drugs? So which means the drug is already available for some other indication, but because XDP is better understood now with research, some drug repurposing is being done. Aloy, please.
So without going into very exact details, no, because all of this is protected, of course, by the institutions and by lawyers, etc. I'm sure everyone understands that. But the idea is with the elucidation of the genetic driver of XDP, including not just the genetic mutation, but also modifiers of disease expressivity, modifiers of transcription, modifiers of how the disease really manifests. All of this was done in genetics with the aid also of cellular modeling. We are able to identify using high throughput based assays, which drugs are able to modify the phenotype of XDP, basing on the signatures that we identified in cells, like the Intron Retention, alternative splicing, and also TAF1 dysregulation. So once you know what to look for, all of this is very easy to run through high throughput assays, wherein you test thousands of drugs and really search for one which will modify the cellular phenotype. Of course, the cellular phenotype is not necessarily always the eventual phenotype. In humans, humans are very different from cellular models. And as we always say, you know, models are essentially useful, but all of them are wrong. But still, you know, there is a benefit and there is a usefulness to using those fundamental studies to come up with pathways and trajectories for translational potential. And one of the things that we're trying to look at right now, instead of trying to discover new drugs, is to find, as Eva said, drugs which have already been out in the market and would just need to be repurposed, because those are, first of all, those won't necessarily have patents, or have patents which are not exclusive to specific companies, but can be used more extensively. And this is something that also relates to the whole idea of using the best resources for a developing nation such as the Philippines.
Thank you, Aloy. That's all, Carlo. Thank you.
Okay, yes, thank you very much. That's very illuminating. I'm glad that we had so many voices speak on the very broad and fascinating topic of precision medicine. And actually, there's still many questions coming in about that, but in order to diversify our discussion, I want to turn to something Dr. Guevara already touched on, which is another question somebody asked to Dr. Domingo and Dr. De Guevara. How is studying of such rare genetic disease done with such small sample sizes? So I think that speaks to concerns about methodology and validity of research methods. No? Dr. Guevara already said a few words there, so maybe Dr. Domingo, you might have some insight in that area.
Yeah, so going back to what I said earlier, I'm a big fan of modeling. Using biosamples has its advantages as well. But I'm a big fan of using genome engineering to model the mutations that you want to study. So in the study which I talked about earlier, some of those mutations are found. So for example, the S21T mutation is only found in individuals in Europe. I think the P54Y, C54Y mutation was described in a patient in China, etc. And I don't necessarily have access to all of those mutations. But what I can do is use CRISPR, put in those mutations in a specific background, and then compare all of those mutations in my own lab and in the assay. So like I said, it has its limitations. These are not samples from the patients themselves, but we can try to isolate what the effects are of the different mutations using modeling. And when I say modeling, it doesn't only refer to the cellular modeling that I do, but Dr. De Castro, Dr. Hibbard also have their own tools for modeling different mutations bioinformatically and also using other methods. But for me, when I deal with mutations which are described singly in certain populations or in different areas of the world, I use genome engineering because for me that's the way to compare all of these mutations to each other.
Thank you very much. Next, we have again some related questions. Apparently, some of our audience members have been inspired to ask about participation or collaboration in these research efforts. So I'll ask these questions together. The first one was addressed to Dr. De Castro. No? He asks, if and how this project accepts volunteers for his research effort. They were impressed with the team welcoming a member from the high school level. And then somebody asked a similar question to Dr. Cochonco de la Paz. They say, I am an MS MBB student at UPD NI MBB, and I'm interested to join the projects related to sequencing the genomes of Filipinos. I would like to ask when these projects will begin in the future and how they can join these projects. And then yet another similar question to Dr. Hibbard asking about collaboration. They ask, what is the best way to maximize the benefits of international research collaboration? So I think there's a level of inspiration that was achieved in this conference. So I'd like to address Dr. De Castro first, and then Dr. Hibbard, and then Dr. De La Paz.
Right. No, thank you. That's a fun question. We had, so Isa is a high school student from UPIS who joined our center during the height of the COVID pandemic. So, I guess people were not doing anything. So there were actually a number of volunteers to the center doing epidemiological modeling, hospital resources modeling, machine learning stuff. And they range from high school students, college students, college graduates, and we even had an MPH student. Well, so we had two MPH students, one from Yale University. She was stuck in the Philippines during the pandemic and was just looking for something to do. So she came by and helped us with a lot of our epidemiological modeling. And then we had also an MPH student from Nigeria who was studying in Angeles. So, we just come across students who are inclined, to help out, and we teach them data work. So mainly we're a center for informatics. So we teach them proper behavior towards data and, how to, a lot of techniques regarding looking for data and analyzing data, visualizing data. These are like very basic stuff that a lot of students are very interested in. We even trained a number of college students in like Southeast Asian competition or national competition. And they were a lot of fun to work with. So we do welcome, we welcome students, all types of students. If they think they can handle data or if they're interested in working with data, any type of data, then you can help us.
Thank you very much. Yes. How about you, Dr. Hibbard? Maybe a few words about international collaboration or participation.
Yeah. So, obviously my heart's in Philippines, but I'm based in London, so that's international collaboration there. And I think essentially international collaborations have to be seen as partnerships. These are two groups with equal contributions. And I think that's an important component of this. And in my experience, the best way of doing this is to have people with like-minded people who are willing to bring forward the new generation and do training as part of the collaboration process. So bringing in the younger people into the partnership is a great way to build international collaborations. And I think that's very important. Thank you.
Thank you very much. Dr. De La Paz?
Yes, I'm going to call on my BFF to co-answer with me. That's Dr. Cynthia. We've been planning the Filipino Genome Project for actually years now. It was one of our flagship programs when we had the next generation sequencers already set up at the Philippine Genome Center. but life events happened, and of course the pandemic happened, and so even if we actually had a good start already sometime in 2020, early beginning of 2020, but then the pandemic happened. And then of course, but we shouldn't feel bad, because by the third day of this conference, you'll hear about the initiatives that are happening now with the population genetics group, and maybe they will be able to demonstrate what they have done so far. But I'd like to ask our I hope I didn't put you on the spot, the executive director Cynthia, but just to show you that this is something that we are serious about, and that it's going to happen. Dr. Cynthia?
So thank you very much for the question. Actually, Dr. Eva and I and other members of the Philippine Genome Center have been planning all along to embark on the Filipino project, because we realize that it's very, very important that we have our own reference sequence, and we know what happened with the sequences in Africa. Even if the original whole human genome sequences were coming from different backgrounds, but until they did the deep sequencing for samples in Africa, they realized that there were several gaps in the genome sequence in the public databases. So in the Philippines, we are also in a very unique position. We have ethno-linguistic groups, at the same time we have populations in urban centers. We did some maths on the sampling sites and the cost of sequencing. So that has been at the heart of the Philippine, because we are the Philippine Genome Center, we have to embark on this very, very important project. We were just waylaid a little bit because of the pandemic, but for sure, Dr. Eva, we are now under pressure. We have to submit that proposal. It has been written in various stages, so what we need to submit it. And I think even the US has already announced that we're going to do Filipino, even if we haven't submitted the proposal yet, because that is how important it is for the Philippines. So that's all. So just listen from time to time for those who would like to get involved in the project. Definitely, we will be hiring people, and at the same time, of course, advising students for this process. Okay, that's all.
Yes, thank you very much. I also just want to add, for those interested in the topic of sequencing of Filipino genomes, please make sure to attend Fred Delphin's talk, which I believe will be on day 3 of this workshop. He'll be talking about the fascinating work that they're doing at NSRI. And with that, now I'd like to touch on 2 questions that relate to technology development and technology transfer. So one person asks, with regards to precision medicine, what is the current status of the collaboration of research institutions with industry, perhaps the startup scene of the Philippines, to develop biotech or biomarker-based products in the diagnosis and prognosis of the mentioned diseases? Is sequencing still the gold standard for these, or PCR-based products? And another person asks, are there typical timeframes for a transfer of technologies, such as the recently emerging nanopore sequencing, from their proprietaries for use in the Philippines? I feel this would be of interest to all speakers, especially in regards to their concerns towards the cost of sequencing as well as data gaps. So broadly, these questions are concerned with the development and utilization of technologies, both sequencing and otherwise, to address our health challenges. And I think these questions could be addressed by all of our speakers. Would anyone like to get that conversation started?
I can see Martin smiling. I think he knows the answer.
I'm happy to start that. Just on the nanopore sequencing. Nanopore is a great technology out of the UK. It's obviously, the cost of the machines is very low, so it's easy to introduce to the Philippines, but the cost of the sequence per base is quite expensive. So it has to be put into the right context and the right applications. around the world, the nanopore is being used, for example, for sequencing the SARS-CoV-2 genome, and that's probably a reasonable application for that. But you wouldn't want to sequence a human genome with the nanopore. So I think a range of technologies are very useful, and I think we should keep abreast of all of those. So part of the London School, we've been busy trying to do training and interpretation of the nanopore data, and we've been using that, for example, on Mycobacterium tuberculosis sequencing, and maybe that's something else that might be a useful tool for this. So we need a range of different genomic technologies, and there's an application for all of them probably.
Okay, I'll
Thank you for that.
continue.
Yes.
So,
Please,
Carlo, I'll
Dr.
continue
Salamat.
with to the person who asked the question. So we are pretty much abreast with the technologies that are coming out of the market. It's not as if we have not heard about nanopore technology. So please erase that from your mind. We are quite familiar with the different technology platforms. Of course, it's a single molecule real-time sequencing system. So nanopore in the beginning was beset with a lot of errors, but that has been sequencing errors. I mean, but that has been to a certain degree improved quite a great deal, and has been some of the workhorse of the COVID-19 sequencing of the different countries around the world. So at the Philippine Genome Center, we also did our crunching of the costing for sequencing, since we already have our own machines and Illumina machines. And we noticed that, of course, the most expensive would be the capital outlay for buying the equipment. Nanopore, so it's not very expensive, probably $1,000 for the min ion. But we already have a lot of Illumina equipment at the Philippine Genome Center, and Illumina to thankfully reduce the cost of the reagents for COVID-seq or for COVID sequencing. So we've been using Illumina. As Martin has mentioned, the machine is not very expensive, but the reagent cost is also expensive. So on cost by cost, or sequence by sequence analysis, when we were doing the crunching for the Philippines, we realized that it's probably a little bit more expensive or just similar to what we have for Illumina. So we retained our workflow with the different Illumina machines we have. Now, in terms of platform, there are many Oxford nanopore technologies, not just MinION, of others, depending on the throughput and the output you want, this Promethean and then GridION. So there are a lot of these technologies, and the beauty of the nanopore technologies is really because the equipment is very, very affordable. Now, in terms of, so you have to use the equipment that would be proper depending on the target you want. So there are, of course, equipment for single molecule sequencing. There are those for short reads that we also do. There are also people who still rely on a microarray technology, and so on and so forth. So there are a lot of sequencing as well as analysis platforms in this regard. So it's very, very important that the complementary technology or the appropriate technology will be used depending on your purpose. And of course, it also is important that we think about cost, turnaround time, etc., etc. And we've been doing a lot of trainings for people, not only for hands-on, but also for bioinformatics analysis. So there's a question, Carla, I'll just finish the question, as to whether or not the sequencing equipment that are coming to the UP Visayas in UP Mindanao in the next 2 weeks will be available also, of course. We decided rather than buying ONT, that we will buy a higher version of Illumina, because that will give the Mindanao a range of opportunities where you can use it for more complex analysis, and of course for human genome, transcriptome analysis, exome analysis that will not be normally affordable using MinION. So that will be very, very useful, and I would like to encourage our researchers in Mindanao to collaborate with our team in Mindanao, because we have a big Mindanao Genomics Consortium. So that will be an opportunity for all of you, and we're excited for the developments beyond COVID of the investments in sequencing technology that we're bringing into the Mindanao region. So that's all.
Excellent. Thank you very much. Unfortunately, we only have time for one last question. And I will take advantage of my moderator position to ask the one that's most relevant to my own work and interests, and that of us here at PGC. No? Somebody asked, what would be the next steps in the genomic biosurveillance of COVID here in the Philippines? So I think Dr. De Castro and Dr. Hibbard might be the ones best equipped to answer this question.
Thank you so much for asking that question. We did, as you can see, we did identify some issues.
Dr. De Castro, if you're speaking, you might be mute. I can't hear you.
Oh, all right. One moment.
Carlo, we can hear
Can you hear
you. We can
me?
hear Dr. John Carlo.
can hear you.
Sorry, I still can't.
Oh, you can't hear me?
The problem
Oh.
isn't on my end, right?
John,
you
we can
can
hear
hear-
you. We can hear you. Go ahead.
All right. Okay, so I'll go ahead and speak. Sorry about that. I think maybe it doesn't want Carlo to hear me. Anyway, we did, as I showed, we did identify some issues with regard to collection submission time lag, which is pretty long for our, for the Philippines. So maybe we can make improvements on that, as well as sampling representation, meaning we should match the amount of sequences that are coming from different geographical areas to the number of cases that are there. And I guess, that's what was coming out from our visualizations. Develop more tools to make quicker assessments of vaccine efficacy if we can, so we can further improve on the accuracy of our predictions. So again, it remains to be seen if we can do that, but we have some initial efforts, and we're happy about it so far.
Okay, thank you very much. Sorry about earlier. I accidentally turned off my volume, so I couldn't hear anyone. Anyway, Dr. Hibbard, what about you? Do you have any insights on advancing genomic biosurveillance for COVID here in the Philippines?
I think the key ingredient is sequencing those viruses. In the UK, we've done particularly well at doing that, through the National Reference Laboratory, and I think that's been giving us an early warning system for understanding which variants are there and how we need to respond to that. So the Philippines is doing well, but I think keeping that up to date is very important. And then we've heard about potential other things that might come through maybe in the near future. One of them is checking for those vaccine resistance ones, and we've heard of a couple of ways that you can analyze your sequences for that purposes. But maybe there will be other tools coming as well, perhaps a severity warning, identifying your patients who might need the treatments. And as we introduce those treatments, those antivirals, for example, which are starting to be rolled out, there might be resistance mutations to those antivirals that might turn up at some point. So I think there's plenty of scope for keeping an eye on this virus, as we move through, and potentially, giving cost-effective interpretation. So this might be reduced to a PCR test at some point, which might be an easier interpretation of the genomic tools. So I think there is scope, and in the Philippines need to keep aware of of what's happening around the world, and keep those technologies available to people within the Philippines.
Carlo,
Thank
can
you very
I
much.
add?
Words to take to heart. Yes, please.
Carlo, I just thought of a couple of things. One is coordination among nations. So, we've shown you the visualization in Southeast Asia, and I guess maybe the next step to that is to really coordinate with our counterparts in the other areas in Southeast Asia, and maybe even the world. We need to have better coordination of our efforts of surveillance, because it doesn't only affect us. For example, if a variant arises in our country, then it also affects the rest of the world in a lot. The other is, and I've actually proposed this a long time ago, we know that, but nobody listened, of course, but we know that COVID is super spread. And so there are only like a very small number of people who contract COVID that actually spread COVID. And this is easily due to the viral load in that particular individual. So one of the things is when one person tests positive for COVID, we don't stop at testing. We need to go and look at the viral load of this individual, because then we can classify which individuals will be spreading the virus versus those, who will not, because they have a lower viral titer. Meaning, if you just do that extra step, it's an easy PCR analysis, then you can tell who can go home, and you can tell who should quarantine. So that's a very quick assessment of.
Can I just answer, Carlo, very, very quickly
Go
about
ahead.
Jong's
Sure.
comments? So actually, Jong, the sequences might not be uploaded in GSAID, but we are fairly current. So we have 17,000 whole genome sequences, but we are not we don't have permission yet to upload because the permission needs to be approved by the Secretary of Health and the Epidemiology Bureau. But as far as the DOH is concerned, we are updated because we share the data to them. But we just want to verify addresses, etc., etc. But they are fairly current. So when they estimate the moment they got a swab around the regions, within 2 weeks, we already know the whole genome sequence. They're just not uploaded in GSAID. So in terms of modeling and mutation analysis, we are also current because there's essentially in the bioinformatics workflow of PGC, it is almost automated. So we also know phylogenetic analysis, and we're running NextTrain internally with all our 18,000 sequences. So, but of course, we are not at liberty to share it with the public. And also in terms of the response, we have the city values, at least all the sequences we have the city values, and the DOH also has that. But at the level of the Philippine Genome Center, we don't look at the patients, we don't have, it is not our part in the whole workflow to be the one engaging with patients and regional epidemiology surveillance units and hospitals. It's really the DOH. So the data that we get, both statistical and modeling, we share to them, and they are the ones who do the outreach to the patients. So that is our role. But we have fairly active data. It's very, very current, except that it has not been uploaded to GSAID until we have received the go signal.
Thank you very much, Dr. Saloma, and to you, Dr. Hibbard, Dr. De Castro, and to all of our speakers and guests that we have today. Unfortunately, that's all the time we have remaining for questions. So thank you to everyone. Thank you to everyone who submitted questions and participated in this talk. Yes, a round of applause to all of you. Thank you very much for everything you did for today. And to everyone watching, if you have more questions about the conference, feel free to email us at capacitydevelopment@pgc.up.edu.ph. And if you have questions about PGC services or research, you can go to our website. Many of our speakers also showed their email addresses at their presentation, so please feel free to reach out to them if you have any further questions or concerns about their presentations. Finally, before we close today's event, I'd like to remind everyone to check the link to the feedback form in our Zoom chat. And it will also be posted right there. It's on the screen. So this is a survey or feedback form. And it will be open until 6 p.m. today. So everyone watching has 2 hours to fill it out. And as I understand it, you need to fill out that form in order to receive your certificate of attendance, if that's something you want to or need to receive for your work. And with that, we conclude day 1 of the PGC Genomics and Bioinformatics Conference 2021 on overcoming challenges and building opportunities. Thank you all, everyone, and we'll see you again tomorrow. So we will now be closing the event, but just a quick word to all of our speakers to please stay. Don't leave the meeting yet, because after everyone has left, we will be having a quick photo session and awarding of certificates. So if you don't mind waiting for a few minutes as everyone else leaves the meeting. To all of our attendees, everyone who took the time this afternoon to watch this conference, once more, I want to thank you very much for your time, for your attention. And we hope to have to see you at future events at PGC. Goodbye, everyone.